all living organisms, both eukaryote and prokaryote, carry nucleic acids as their genetic blueprint. Viruses also contain nucleic acids, but they cannot reproduce on their own. Instead, they must invade a living cell because they lack ribosomes and other metabolic machinery. Viruses are obligate parasites. They reproduce only inside a host. Viruses infect every form of cellular life and show astonishing variety in shape and structure. Scientists classify them by the type of genome they carry and by the way they replicate. In this lesson, we're going to understand their shared features, their structural diversity, their life cycles, and the evidence for their origins and rapid evolution. Viruses can be grouped first by the type of nucleic acid they carry, DNA or RNA. Among DNA viruses, some are single-stranded and others are double-stranded. A classic single-stranded DNA virus is a bacteriophage, M13, about 500 nanometers long. Herpes simplex represents the double-stranded DNA group. It infects animals and is roughly 200 nanometers across. RNA viruses also appear as single or double-stranded particles. Poliovirus is a single-stranded RNA virus in animals, barely 25 nanometers wide. Human immunodeficiency virus, or HIV, is another single-stranded RNA virus. As a retrovirus, it first copies RNA into DNA. Reovirus typifies double-stranded RNA viruses, measuring roughly 80 to 100 nanometers. These examples appear in Figure A 2.3.1, highlighting how genome type drives viral classification. Remember, viruses are excluded from both the Linnean hierarchy and the three-domain system. Unlike cellular life, which traces back to last universal common ancestor, viruses can have multiple independent origins. For that reason, virologists often sort them by genome, DNA or RNA, single or double-stranded, and for RNA, positive or negative polarity. Some infections are caused by microorganisms like bacteria. Others are caused by viruses, but bacteria and viruses are fundamentally different in both structure and life cycle. In the table A2.3.1, we can outline the key differences between bacteria and viruses. Okay, so here we have the table viruses versus bacteria. Are they cellular? Viruses are not. In terms of size, viruses range from 0.02 to 0.2 micrometers. Bacteria are much larger, about 1 to 5 micrometers. Nucleic acids. Viruses carry either DNA or RNA, but never both. Bacteria have both DNA and RNA. According to the ribosomes, we can say that ribosomes are rare in viruses. But ribosomes are present in bacteria as 70S ribosomes. Outer surface. Viruses may have a protein capsid and sometimes a lipoprotein envelope. Bacteria have a cell wall made of peptidoglycan. Motility. Viruses don't move. Some bacteria do, using rotating flagella. Replication. Viruses rely on host cells. They use protein synthesis and self-assembly. Bacteria reproduce independently through binary fission. To sum up, Viruses and bacteria are both microscopic, but behave in completely different ways. Science is based on observation and testing ideas. Before seeing viruses, scientists predicted they existed. They used filters to remove bacteria from sick tissues, but the infection continued. That meant something smaller than bacteria. In 1915, Frederick Lord discovered bacteriophages. They are viruses that infect bacteria. In 1798, Edward Jenner created the first vaccine against smallpox. He used fluid from cowpox to protect people. Louis Pasteur later developed a rabies vaccine. Even without seeing viruses, scientists learned to fight them. In 1966, the ICTV was formed. It classified and named viruses. For example, HIV belongs to the Retroviridae family. The goal is to describe and organize every virus. All viruses are small. They have DNA or RNA, as we previously saw. They have a protein code called a capsid. They have no cytoplasm. They have a few or no enzymes, and they do not have a nucleus or a nucleoid. Viruses are highly diverse. They differ in genome type, shape, and replication. Some viruses are spherical, others are complex. Some have envelopes from the host cell membrane. Viruses are classified by genome and replication. The ICTV uses taxonomy like in biology. The Baltimore system groups viruses by how they replicate. It includes DNA, RNA, and retroviruses like HIV. Viruses changed science before they were seen, before they were even seen. They are small, invisible, and powerful, and they are still shaping the future of biology. Now we'll explore the structure of viruses, how they replicate, and what makes them unique among biological entities. Viruses are microscopic infectious agents. They don't perform life processes on their own, but hijack host cells to reproduce. DNA viruses include adenoviruses and most bacteriophages. RNA viruses include HIV and coronaviruses. SARS-CoV is a virus that causes COVID-19. Positive sense RNA acts like messenger RNA and can be used to make proteins. Negative sense RNA must be converted to positive sense before use. Some viral genomes are linear like coronaviruses. Others are circular like hepatitis B. Genomes may be single-stranded or double-stranded. The capsid is a protein code protecting the viral genome. It's made of subunits called capsomeres. Shapes include helical, conical, and polyhedral forms. Enveloped viruses steal parts of host cell membranes. These contain spike proteins to help enter host cells. Viruses reproduce through steps: attachment, entry, replication, protein synthesis, and release. Some follow a lytic cycle, others a lysogenic one. HIV uses reverse transcriptase to make DNA from RNA. Coronaviruses existed before COVID-19. SARS-CoV-2 was identified in 2019. Viruses are not alive, but they interact with all life. Their genomes are small, efficient, and perfectly adapted to replicate fast. Bacteriophages are viruses that infect bacteria. Lambda-phage only infects E. coli. Its structure includes a capsid, a tail, and tail fibers. The lytic cycle destroys the host cells. It starts with the phage attaching to the bacterium, then it injects its DNA and replicates. New phages are assembled and the cell bursts. In the lysogenic cycle, the viral DNA becomes part of the bacterial DNA. This viral DNA is called a prophage. It is passed on when the bacteria reproduced. Stress can activate the prophage to enter the lytic cycle. Viruses may have evolved from escape genes or from ancient cells. They evolve rapidly due to mutations and fast replication. RNA viruses mutate more than DNA viruses. Now let's talk about mutation drift and shift. Antigenic drift causes small changes in surface proteins. Antigenic shift causes big changes through recombination. Okay, here we have a case study HIV. HIV is a retrovirus that RNA uses to make DNA. It integrates into host DNA and becomes a provirus. Mutations make it hard to treat with just one drug. Thanks for learning with us. Viruses are tiny, but their impact is huge. Like and subscribe for more science videos here to Lingually International and Michelle Wright again. Hope to have helped you a lot.